Hi, I'm Bethany Cummings. I'm an assistant professor in the Department of Biomedical Sciences at Cornell University, and today I'm going to talk about my lab's publication in Gut entitled TGR5 Contributes to the Glucoregulatory Benefits of Vertical Sleeve Gastrectomy Surgery in Mice. The prevalence of obesity and type 2 diabetes has reached epidemic levels worldwide. Therefore, there is an urgent need for the development of novel therapeutic strategies for these diseases. Bariatric surgery is defined as a surgical manipulation of the gut that's performed with the goal of producing body weight loss. Bariatric surgery is the most effective long-term treatment for obesity and results in high rates of type 2 diabetes remission. Importantly, the effect of bariatric surgery to cause type 2 diabetes remission is often observed soon after surgery prior to significant body weight loss. This suggests that there are body weight independent mechanisms by which bariatric surgery causes type 2 diabetes remission. So the overarching concept is if we can define the mechanisms by which bariatric surgery causes type 2 diabetes remission, we may be able to identify novel therapeutic strategies for type 2 diabetes treatment. In order to accomplish this, my lab has developed a mouse model of vertical sleeve gastrectomy surgery, or VSG for short. VSG has become the most commonly performed bariatric procedure in the clinics in the US. It's performed by removing the majority of the stomach by cutting along the greater curvature. This is how the procedure is performed in humans and how we perform it in our mouse model. We also perform a sham surgery to control for the effective surgery. Sham surgery is performed by placing suture in the area that would be otherwise transected. Many mechanisms have been suggested to contribute to the glucoregulatory benefits of bariatric surgery, including bile acids. Circulating bile acid concentrations go up after various types of bariatric surgery, including VSG. TGR5 is a transmembrane G-protein coupled bile acid receptor that has been implicated in the metabolic benefits of bile acids. Therefore, we hypothesized that TGR5 contributes to the glucoregulatory benefits of VSG. We tested this hypothesis by applying our mouse VSG model to a whole-body TGR5 knockout mouse model. We had 6 study groups. We had sham-operated mice that were fed ad libitum. We had sham-operated mice that were food-restricted to match their body weight to the VSG-operated mice and therefore control for the effective body weight. And then we had a VSG-operated group. And we studied these 3 groups in both the TGR5 wild-type and knockout condition. Consistent with what we see in human populations, VSG decreased food intake and body weight, and this was independent of genotype. VSG also improved glucose tolerance, independent of body weight, and this effect was lost in TGR5 knockout mice. As expected, VSG increased total circulating bile acid concentrations, and this was independent of genotype. However, VSG also produced metabolically beneficial shifts in the circulating bile acid profile that were dependent on TGR5. Specifically, TGR5 promoted shifts in the bile acid profile that have been associated with improved insulin sensitivity in humans. Therefore, the effect of TGR5 to improve glucose regulation after VSG may be due to its effect on bile acid profile. 2 major determinants of bile acid profile are hepatic bile acid synthesis and bile acid metabolism by the gut microbiota. Therefore, we measured the expression of hepatic bile acid synthetic enzymes and found a TGR5-dependent decrease in hepatic CYP8B1 expression. Furthermore, we measured by 16S rRNA sequencing gut microbial profiles and found a TGR5-dependent shift in gut microbial profile after VSG. Therefore, the effect of TGR5 on hepatic bile acid metabolism and possibly on gut microbial bile acid metabolism may contribute to its effects on bile acid profile after VSG. So to summarize, what we found is that TGR5 contributes to the glucoregulatory benefits of VSG. Furthermore, we've identified a novel role for TGR5 in the regulation of bile acid metabolism. Further work is needed to understand how TGR5 regulates bile acid metabolism and how TGR5's role in bile acid metabolism may impact whole body glucose metabolism.